Hi, and welcome to this run-through of the AQA A-Level Biology Paper 1 from 2019. Now, the reason I'm doing 2019 and not the 2023 paper is because actually if you're sitting the exam in 2024, probably the best papers for you to be doing as you get closer and closer to the final exam are the papers 2019, 2020, and possibly 2018 as well. That's because these papers, they were over 3 years ago, and it's more likely that the stuff that was in them might be similar to the stuff that's going to be in the papers this year. So these papers are really, really good ones to practice. So let's get into it. All right, first question. Okay, so this first question here, describe how a non-competitive inhibitor can reduce the rate of an enzyme-controlled reaction. I've dropped in a little picture here. This is the sort of thing that you should have in your head, this image for the non-competitive inhibitor. The key thing is the inhibitor binds to the protein, to the enzyme, somewhere other than the active site. You can call it an allosteric site. And when it binds to this other site, it slightly changes the tertiary structure of the enzyme, changes the shape of the active site, so that now the substrate can no longer fit into the active site. Okay, so that's what you need to say. Let's double check the mark scheme to check if there's any really, really specific language that you need to use. Attaches to the enzyme at a site other than the active site. You can say allosteric site. Changes the shape of the active site or tertiary structure. I've seen a lot of mark schemes actually are even more specific. They require you to say tertiary structure, and sometimes they don't even accept shape. So that, okay, I'm not sure if I would have said this last thing, would I have got the mark? I said, so the substrate can no longer fit or bind. So I think I would have missed this mark. I'm still relatively new to AQA. Complementary is a key word you would have to say, so that the substrate is no longer complementary. Okay, moving on. Okay, so here we have a graph. Pectin is a substance found in some fruit and vegetables. It actually is a glue that holds the cell wall together, the different cellulose fibers together. A scientist investigated the effect of pectin on the hydrolysis of lipids by a lipase enzyme. Here are the results. So we've got the lipase activity on the y-axis, and we've got the lipid concentration, which is the substrate concentration, on the x-axis. Now this graph should scream non-competitive inhibitor for you, because what you kind of need to remember that if pectin was a competitive inhibitor, not a non-competitive inhibitor, then the graph would be looking more like this and would eventually reach the same level as without any inhibitor. So this really should scream non-competitive inhibitor down here. The scientists concluded that pectin is a non-competitive inhibitor of the lipase enzyme. So how do you explain that? How do you explain what I've just drawn in words? You would say that adding more and more substrate, that's lipid, does not change the maximum rate of lipase activity. So let's see if that's exactly what the mark scheme has said. So with the inhibitor, increasing substrate, so adding more substrate, does not increase or change the rate of reaction. Yeah. Or high substrate does not overcome the inhibition. What that's talking about, this overcoming the inhibition, if we go back to this little picture up here, with a competitive inhibitor, which isn't what we're looking at in this question, but with a competitive inhibitor up here, if you add more and more substrate molecules, more of these blue molecules, if you add enough of them, there'll be so many that they are more likely to enter the active site than any inhibitor. So like if you have 99% substrate and only 1% inhibitor, most of the time substrates will enter. Okay, let's keep going. The scientists also found that pectin stops the action of bile salts. He prepared two suspensions. Suspension A has lipid and bile salts, and suspension B has lipid and bile salts and pectin. He did not add lipase to either suspension. Okay, so we need to kind of spot this, bile salts, and what is the function of bile salts. So the function of bile salts is that it emulsifies fat. Okay, and that's the key thing. Now, what does that mean, emulsifying fat? Emulsifying means taking larger droplets and breaking them into smaller and smaller droplets. That's helpful because it increases the surface area available for digestion, and we're going to see it potentially in this picture. Okay, so in this picture here, we can already kind of see that we're getting a result that we would expect, because in this picture here, I can't see anything, and in this picture here, I can see droplets. So potentially it is the fact that the pectin is somehow interfering with the emulsification of the fat droplets in suspension B. Let's have a look. Let's have a look at the next question. Okay, so the formula you need to remember is contained in this kind of I-AM triangle. That's how I say it. Or AIM triangle, so actual, image, and magnification. You can draw it like this if you want to. So what we're looking for is the actual length of this droplet, which is the image divided by the magnification. So image divided by magnification. Now the image, I can't measure it on my screen, but let's just call it L for whatever length you measure it in millimeters, and divide it by the magnification, which is given here, 500. So that's going to give you an answer in millimeters, but you need to spot the fact that you want to give the answer in micrometers, so you're going to have to times that by 1,000. There are 1,000 micrometers in a millimeter. Right, what about this a bit about uncertainty? Okay, so this uncertainty bit, I must confess, it really threw me. I'm still new-ish to these uncertainty questions because they don't really come up in OCR, but they do in AQA. So the thing that you needed to spot here was this: using a ruler with millimeter intervals. So what kind of uncertainty is this going to generate? So if you're measuring something with a ruler, you're actually not making one measurement, but you're making two. So that's shown down over here. I've just dropped this little image in. So there's a bit of an uncertainty to do with where your start point is, where you're using the ruler, and an uncertainty with where your end point is. So therefore, the uncertainty is actually equal to 2 times like half the smallest measuring increment. So the smallest measuring increment is a millimeter. So that means you could, each time you're measuring, you might be like up to half a millimeter out. So it's 2 times 0.5 millimeters is the uncertainty, so therefore it's 1 millimeter is the measuring uncertainty. So then if we look at the measurement and the calculated maximum length, we've got to do the same thing that we did before. So if it's 1 millimeter is the uncertainty, but we want it in micrometers, then what we need to do is, first of all, divide it by 500, but then we need to times it by 1,000 to convert into micrometers, and you'll see that that is 1 divided by 500 times it by 1,000. That's basically times it by 2. So that is 2 micrometers is the uncertainty of your calculated length. Okay, so the key thing to remember there, if you're using a ruler, it's actually 2 measurements, not 1. All right, let's move on. 1.4. It says, no large lipid droplets are visible with the optical microscope in the samples from suspension A. We already talked about it a little bit, and we said that we think that that's because it's been emulsified. But where's the second mark? So when reading this question, no large droplets are visible with the optical microscope. The question highlights the fact that this is an optical microscope, and what do we know about that? It has a lower resolution. So 2 things to say. That the fat droplets have been emulsified, which means kind of made into smaller droplets, and now the resolution of the optical microscope is not sufficient to see those smaller droplets. Let's check the mark scheme for those 2. Here we go. So first of all, that maximum length, there is a bit of tolerance there, 8 to 10 micrometers, and the uncertainty is 2 micrometers. For the droplets question, I've got 1 mark for emulsification, and the second mark, you have to use the word resolution, okay, due to the resolution. You cannot be seen due to the resolution of the microscope. You could get a mark, it does say over here to the right, if you're talking about the wavelength of light. So the wavelength of light is too large to be able to see the droplets. You could have talked about it that way, but maybe easier to just say resolution. All right, let's move on to question 2. Okay, question 2. So the first part is just about what things would you find in the cell wall of different types of cell. So we've got plants, algae, fungi, and prokaryotes. What would we find? So you need to remember that, hopefully you remember that plants have cellulose in the cell wall, and that is also common with algae. Fungi, you should remember that it's chitin, and prokaryotes, you should hopefully remember that it is murine. So this murine, you might also sometimes see it written as peptidoglycan, or that, though more commonly that's used in OCR, but peptidoglycan is the same thing as murine. Right, let's keep going. Cell walls make up much of the fiber that people eat. That's always a good idea to kind of highlight things as you go, things that you think are going to be relevant. Scientists investigated the relationship between mass of fiber people eat each day and their risk of cardiovascular disease. So relationship, that's something that, maybe a word that kind of flags up relationship. So we're talking about correlation here. They gathered data from a large sample of people. That's a good thing. Maybe it might be worth marks later on because this is like indicates that this data might be valid. And use this to calculate a relative risk. Okay, so this is an interesting term, relative risk. What does this mean? You should probably be familiar with this generally, as I've seen it come up a couple times. So a relative risk of 1 means there's no difference in risk between the sample and the whole population. A relative risk of lower than 1 means CVD is less likely to occur. And a relative risk of bigger than 1 means CVD is more likely to occur. So remember, just in terms of your math skills, you do need to know the meaning of these symbols, greater than, smaller than, so on. Okay, here are the results, the results are shown in figure 3, and it also says a value of plus or minus 2 standard deviations from the mean includes over 95% of the data. So because we're looking at standard deviations here, I know that there's going to be some marks later on for talking about whether a difference is significant or not. Because remember, if the standard deviations overlap, it's not significant. And if they don't overlap, it is significant, more likely to be significant. Right, let's have a look. First of all, I'm already going to look at this graph before I even look at the question, just to kind of get a feel for it. And I might just look at this value here, okay? Because that's where we've got a higher risk than normal and a lower risk than normal with our fiber intake. And it looks like the key value there, I think, is 18 grams of fiber seems to make the difference. Okay. It is an evaluate question, of course it is. So let's look at the statement carefully first. A student concluded from figure 3 that eating an extra 10 grams of fiber per day, that's interesting, an extra 10 grams of fiber per day would significantly lower his risk of cardiovascular disease. So 2 things to pick up there. First of all, this is weird that it's an extra 10 grams, not going from 0 to 10 or going from 20 to 30, but just an extra 10. And then, is it significant or not? Okay, so we want to be looking at error bars. Another thing, this student is saying, if I do eat this fiber, it will lower. So that is that this would is kind of to do with cause, but remember, this is maybe not cause, this is correlation. So there's some things to pick up in the sentence there. All right, so when looking at this evaluate for marker, we're going to want to try and have some things for and some things against, maybe 2 points for and 2 points against. And you might go as far as trying 3 for and 3 against, just to make absolutely sure you get all the marks you deserve. So let's go with the easy one first, the for. Remember, this is talking about correlation. So this graph shows correlation, doesn't it? It says, as the mass of fiber consumed a day increases, the relative risk of cardiovascular risk decreases, showing a negative correlation. So negative correlation. So I would probably say negative correlation and describe it, just to make absolutely sure. And then what else? What else? Other positive things to say about it. Okay, so we could say where on this graph might eating more fiber give you a significant result. Let's say if we started, I'm going to zoom in a little bit. Let's say if we started at a position of here, which is 6 grams of fiber eaten a day, and we went to 16, which is here, right? That is a significant decrease. This is the error bar there. But now there's basically no error there. It's like super tight. Okay, so this would be a significant decrease. Sorry. So that's something you could say for the positive. So 6 grams up to 16 grams, significant. But actually, it was a bit difficult to find another thing for pro this statement. It's probably easier to find things against this statement. So against the statement. Well, let's start with the same sort of point we just made. What about if you were down here? What if you were sort of already eating, let's take this data point here. No, let's just go there. Let's say if you were eating 40 grams of fiber a day and then you went up to 50 grams of fiber a day, that's 10 extra grams, right? Well, what's that going to do to you? It's going to do nothing really, because there's the error bar for that one and there's the error bar for that one. So the error bars does produce a small drop, but the standard deviations overlap a lot and there is no significant reduction. So it might even be worth 2 marks for saying that. So 40 to 50 grams, standard deviations overlap. That might even be worth 2 marks. We should definitely say something else. The other thing I would say is this cause idea, causal. So it's not necessarily causal. So correlation is not causation, a standard phrase you could always say. And another thing that you could throw in as well, if you just want to get an extra mark, hopefully, sometimes it's there, is there's no statistical test done to see if it is, well, statistically valid. So there has not been a statistical test. Let's look at the mark scheme for this question. Okay, so first of all, in terms of the marking of this question, the marks are grouped like this. So these 2 columns equal a mark 1. This is another mark 1, and this is another mark. So there's 3 marks for that. Let's look at the evaluation. All right, so first of all, let's highlight in different colors because it's sometimes difficult to kind of understand. So let's do points for the statement. The fact that there is a negative correlation, we have to have the word correlation. Okay, and then this so point 4, you could either kind of have for or against. So if we went between 5 and 30 grams a day, so if we talked about towards the left of the graph and gave data that indicates a significant drop, that would be worth a mark. You can't get the mark twice. Let's use yellow for against. So you could also say it between 30 and 50. So if we said 40 to 50 grams, that's not going to give a significant increase. It's worth, I didn't say this was an obvious thing to say. The student claims that eating 10 extra grams of fiber will give them a lower chance of CVD, but we don't know what their current fiber intake is. Maybe they're already at 40 grams. We don't know. Maybe they're at 0 grams, in which case it will help, but we don't know. I guess if you just mentioning this idea is worth a mark. Okay, either for or against, mentioning the fact that the standard deviations either do overlap or don't overlap, and linking that to significance is credited. Other stuff, we said correlation does not mean causation. Same mark point as saying another named factor may be involved, for example, whether they smoke. So there has to be a named factor. So you could say something with more significance might be whether this person smokes or takes regular exercise. Interesting. What else do they have? Little evident. There's not many data points for higher masses of fiber. There's large standard large standard deviations either at the beginning of the graph with low mass or at the end of the graph with high mass. And remember we said that this one often is there. If you're struggling, just throw this in. There's no statistical test done. Okay, so gosh, there's lots of different things you could have said there. I'll zoom out slightly, and if you want to have another look at those, pause and reflect on what other things you could have said. Okay, let's move on. The scientists estimated the mean mass of fiber eaten per day using a food frequency questionnaire. Interesting. The FFQ asks each person how often they have eaten many types of food over the past year. An alternative method to calculate fiber eaten is for a nurse to ask each person detailed questions about what they have eaten in the last 24 hours. Okay, this is strange. Okay, so just thinking about this out loud. So an advantage of FFQ and one disadvantage of FFQ. So FFQ is the over the past year. So the advantage is the fact that your diet is going to be different every single day. So by asking people to estimate what they've eaten over the past year, you're getting more of an average view of the diet. Because some days, maybe you eat fish and chips on a Friday, every single Friday, but then you eat some salad on a Monday. I don't know. So maybe it changes. Disadvantage is obviously that I cannot remember what I've eaten accurately over the past year. I'm not sure I would be able to give a sensible estimate of what foods I've eaten and how frequently I've eaten them. I just think it would be way off, to be honest. So let's see what the mark scheme says. Would I have got marks for saying that? So okay, so yeah, the fact that diet may vary during the day and therefore it's more representative over a longer period is basically all the same mark point here. And then disadvantage relies on long-term memory, so may not be accurate. There's lots of other things you could have said. But that's what, recall of 24-hour diet likely to be more accurate. I can remember what I ate for lunch and what I ate for dinner last night. Beyond that, it's a bit blurry. So yeah, those are your possible answers. Okay, on to question 3. This says, a group of students investigated biodiversity of different areas of farmland. They collected data in each of these habitats: the center of a field, the edge of a field, and a hedge between the fields. So if you don't know kind of what the English countryside looks like, you can't picture this, I'm going to drop a little picture in here. There it is. So in the center of the field, we've mainly got the crops that the farmer is planting. Around the edge of the field, we're quite close to the hedges. So this data kind of matches what I would expect, and the hedges are kind of a very thin but wild habitat where we'd expect more biodiversity. So the first question is, what data would the students need to collect to calculate their index of diversity in each habitat? So for any index of biodiversity, we're kind of looking at both the species richness and the species evenness. So that means that you need to count the different species that are present and also the number of each individual species in order to calculate biodiversity. Let's double check the mark scheme, but that should probably be it. The number of species and the number of individuals of each species. Okay. The follow-up. Give 2 ways the student would have ensured their index of diversity was representative of each habitat. So representative is the key word here. So this is about making sure that you have enough samples and they're taken in the right way. So sufficient number of samples, so large number of sample sites in each of the areas or each habitat. Also that the sample sites would be chosen by randomly. You could say use a random number generator to identify sites for sampling. Let's just move on to 3.3 before we look at the mark scheme. Modern farming techniques have led to larger fields and the removal of hedges between fields. Use figure 4 to suggest why biodiversity decreases when farmers use large fields. Perhaps I can illustrate here. Okay, here is a big patch of green space, and then let's say that we've got hedges down the middle. So this is like 4 fields with hedges all the way around. Okay, what happens if we remove some of these hedges? Well, if I remove this hedge here and then this hedge here, now I have much fewer, well, a lot less hedge. I've got a lot less hedge, and I've also got a lot less, excuse me, edge. Okay, because the bar chart above says that the edge regions close to the hedges are also sites of high biodiversity, so we're losing that as well. So that's going to reduce biodiversity quite a lot. The mark scheme. So first of all, how can we make sure the sample is representative? We're looking for random samples, and we're looking for a large number of sample sites. This way of saying it is interesting. I doubt anyone would have said this, but basically if you keep doing sampling and you're working out an average biodiversity, and then if you keep sampling more and more and more, eventually your biodiversity index is just not going to change. It's going to be very close to the kind of true value of biodiversity. That's what that means. 3.3, the mark scheme down here. As you have larger fields, you have more center or less edge, basically. Less hedge, and therefore fewer species. This is interesting over here. So if ever something in a question, if ever something is said in a question, you can't just repeat it. Okay, it says ignore removal of hedge as given in the stem. That means in the question stem. You can't repeat something that they say in a question and expect to be given credit for it. Okay, moving on with more question 3. Farmers are now being encouraged to replant hedges on their land. Suggest and explain one advantage and one disadvantage to a farmer of replanting hedges on her farmland. Well, let's actually go back to this picture up here. First of all, if I were to take this large field and replant a hedge down the middle, going back to this one, well, look what I'm doing. I am removing a small portion of the area of the field. So that is, there's going to be less area for farming crops, so that's a disadvantage. But what about the advantage to and it says to the farmer. It's not the advantage to biodiversity, it's the advantage to the farmer. Well, what we find actually is that living in the hedges and also the areas around the hedges are natural predatory insects and things like spiders, that can actually prey on any pests in the field. So that might be an advantage. Predators can act as natural pest control by kind of moving from the hedges back into the field. There'll be some other ones. Let's look at the mark scheme. Okay, so advantage. I think, okay, so this is interesting. We would actually need to say not just an increase in predators to prey on pests, but we actually have to say a greater biodiversity, so increase in predators of pests. Maybe this would actually be credited. So increase in predators, so there's more yield. So we have to link two ideas together. Look at all this so. So increase in predators of pests, so therefore higher crop yields. Increase in pollinators, so higher crop yields. This is pretty abstract down here. I'm not sure about that one. The main disadvantage, the one I said, was reduced land area for crop growth. Isn't it interesting here that in this mark scheme, you can say the opposite thing and get credit for it. So this says increase in predators, and this says increase in pests, but they're kind of both credited. You could argue, well, it could be an advantage or a disadvantage. Just to zoom out a little bit there so you can see the full picture. Pause and have a look at some of those other possible options, and then we'll move on. Oh, I just actually spotted this down here. More difficult to farm. This is almost like an economics angle. It's kind of interesting to see this in the mark scheme, but I was thinking this. It's harder to get a tractor between multiple small fields. If you remove the hedges in the middle, the tractor can just plow up and down. It's more efficient. So that's kind of an economics angle, and that is actually one of the reasons why a lot of farms have removed hedges, is because of the economics of tractors. All right, I think we're on to next question. Yes, here we are. Question 4. Okay. Scientists collected data on 800,000 human births. The data showed the mass of each baby at birth and whether the baby needed to be transferred to a special care unit for very ill babies. Okay, so this here shows the mass at birth in the middle, population frequency, and the transfer to special care unit. Now, hopefully, when you see this graph, you should basically automatically be thinking stabilizing selection, because this is one of the key examples of stabilizing selection, birth weight of human babies. What this shows is that babies who are born of very low mass at birth, let's say 2 kilograms, 2,000 grams, they are much more likely to need to go to a special care unit, and therefore they are more likely to die. And also babies that are very large, so 5 kilograms, for example, is the same story for them. I guess this right here in the middle is the mode, and I suppose probably also the median birth mass looks to be, I think, 3.15. Yeah, 3,150 grams looks to be the mode birth mass there, and it's pretty close to the median mass as well. Okay, so use figure 5 to explain. Okay, great. Use figure 5 to explain how human mass at birth is affected by stabilizing selection. So we basically just kind of explain in a bit more detail what I've just said. Babies of low birth mass are more likely to go to the special care unit and therefore are more likely to die, less likely to survive and pass on their genes. Same thing for the bigger babies. So this means that extreme phenotypes are removed from the gene pool, and this is an example of stabilizing selection. Let's check the mark scheme to see if there's any more specific things you need to say. Most likely. So this is what we said. Extreme mass babies are least likely to survive, and so less likely to pass on their... I said genes, didn't I? Alleles. Got to say alleles. Less likely to pass on their alleles rather than genes. Okay. When talking about natural selection, you've got to say alleles, and I need to remember that as well. Extreme mass at birth decreases in frequency. So change in the allele frequency. We could say the allele frequency for very low or very high birth masses decreases. And this is an example of stabilizing selection. Interested that you have to say, it does say, okay, most likely to be transferred are those under 2,800. Over here it says, except any value between 1,400 and 2,800. And for the big ones, any value between 4,200 and 5,200. I want to look at that graph again, because that's kind of interesting. So is it from 1,800, they said? They said between 2,800. Okay, basically they're talking about, okay, I understand now what they're talking about. They're talking about you're just basically describing this part of the graph down to there, and this part of the graph down to there. So basically the idea that, if you're in this yellow region of birth masses, is highly unlikely that you're going to need to go to any special care unit. It doesn't seem to have much effect whether you're a little bit above or below this median, but if you then go outside of this yellow range, then the risk of going to the special care unit then becomes a lot higher. Okay, right, let's keep going. On to question 4. That was a short question. Okay, the scientists study the effect of one form, KR2DS1, of the human KIR gene on mass at birth. Oh no, this is still the same question. In the following passage, the number of spaces can be filled with biological terms. Okay, so let's just go through it. KRI2DS1 is an allele of the KIR gene found at a locus. Okay, this is where we put somewhere. This is where a place where a gene or an allele of that gene is on a chromosome. Found at a locus on chromosome 19. Blah, blah, blah is 14,021 bases long and is something into mRNA, that is blah, blah, blah. So DNA to RNA is transcribed. This mRNA is then something that's translated into a polypeptide, 304 amino acids long. The polypeptide is then modified in the organelle, the Golgi body, Golgi apparatus or Golgi body, before forming its functional. We don't know if it's quaternary, so I'd probably say tertiary. Oh no, it says modified actually. Hang on a minute. Maybe I'd say quaternary. Oh, 50-50. Hopefully they'd accept both. I would probably say quaternary because it's modified, but I might be wrong on this. Let's have a see if I'm right or wrong. Oh, I was wrong. Should have said tertiary. So this is right. Allele, locus, transcribed, translated, Golgi, or the rough endoplasmic reticulum, either one. I should have said tertiary there. Tertiary is the right answer. I thought they were being tricky with it. Tertiary. Moving on. Okay, still on question 4.3. The scientists studied 1,500 more births, in addition to the ones they studied earlier. They recorded the mass at birth of each baby and the nature of the KIR gene in the mother's genome. Okay, so between 2,500 and 4,500 grams. And this is, more absent. And this is still more absent, but the ratios are a lot different. It might be worth looking at the ratios here. Maybe, but I'll keep going and we'll see if we need to do that later. The scientists used a statistical test to test the following null hypothesis. The presence of KIR in the mother's genome does not affect the frequency of births above 4,500. Does not affect. Tick one of the boxes that gives the name of the statistical test that scientists should use with the data. Okay, well, the one thing, actually this is easier than I initially thought. There is no mean here. There is no standard deviation, so we can't use student's t-test. We're not looking at two different variables, so it's not correlation coefficient. It's chi-squared, kind of gives it away. So how would we actually use chi-squared in this scenario? I don't think the question asked us to do it, but how would we? I think we would have to look at the total. Okay, so yeah, I think what we would do, this is kind of beyond, but what you do, I think, is you'd have to add these two to get the totals for that, totals for present, totals for absent, and then use those totals to kind of work out an expected ratio if there was no impact. So it's like expected ratio of these if there was no impact, an expected ratio of these if there was no impact, and then do the observed minus expected to see if our observed values are statistically different than the expected values. Luckily, we don't have to do that in this question because I think the next bit just goes into p-values. Okay, so the scientists calculated a p-value of 0.03 when testing their null hypothesis. So this means significant. P-value, it is lower than 0.05. So that means that there is only a 3% chance, right? There's a 3% chance that the data here, that this data here could be is due to chance. There's a 3% probability that the difference is due to chance. So therefore, we reject the null hypothesis, and we accept the hypothesis that this allele, presence of this allele, does affect whether the baby has an extreme birth mass. Did I get 3 marks? I think it's probably worth saying the p-value is below the critical value, below the 0.05 critical value. Oh yeah, I think I would have just about got the marks there. We have to say probability. Look at this. There's a lot of underlining here, which means that the way you need to say it is very specific. The probability that the difference is due to, there's 3 underlined things here. The probability that the difference is due to chance is less than 0.05. Or you could say the probability that the difference is due to chance is 0.03. So therefore we reject the null hypothesis, and the allele does affect the frequency of high birth mass. Okay, so a p-values question like this, the wording is always going to be the same. There's ever another p-values question like this, so you basically can just learn this wording. So definitely worth doing. P-values does come up again and again and again. Okay, on we go. Here we go. New question. Describe the structure of the human immunodeficiency virus. So I thought I'd put in a little picture over here so we can just talk about it. Here is the structure of the human immunodeficiency virus. Starting from the outside, we have a lipid envelope. That's actually derived from the cell membrane when it kind of pops out of it. Beneath that, we've got this protein capsid. So that's this blue bit around here. Inside the protein capsid, we have the viral RNA. So RNA is the genetic material. We also have reverse transcriptase enzyme present because that catalyzes the formation of DNA from the viral RNA. Let's check the marks. Okay, 5 different things you could say. RNA, reverse transcriptase, the capsid, lipid envelope, or envelope made of membrane. I didn't say attachment proteins. There are also attachment proteins there. Forgot to highlight those. Where are they? Yeah, these are these kind of spiky bits, the purple bits that stick out, that protrude from the membrane. So those are the marks. You might have described 5 as a glycoprotein. Pause if you need to, and then let's continue. Some people infected with HIV do not develop AIDS. These people are called HIV controllers. So remember, HIV is the virus and AIDS is acquired immunodeficiency syndrome. So this is like being symptomatic and having a reduced immune system, basically. Scientists measure the number of HIV particles, that's called the viral load, and the number of one type of T helper cell, called a CD4 cell, in the blood of a group of HIV controllers and also in a group of HIV positive patients who had symptoms of AIDS. The median values and the range of the results are shown in the table. Okay, this is important. So median and the range. So the median viral load and then the range in brackets. So, right, let's look at these numbers. So for HIV controllers, the viral load is way lower, 212 to 66,274. And actually also, if we look at the range of the HIV controllers, the highest person of all the HIV controllers had a viral load of 609, and that is significantly lower than the lowest person here. So this is like kind of showing me, it's not error bars because it's not standard deviation, but the range bars are way not overlapping. What about the CD4 cells? Let's look at that. 693 to 248. Now there's still no overlap. The lowest number of CD4 cells, because CD4 cells are good and we want lots of them. So the lowest number of CD4 cells in the HIV controllers is 529, and the highest in the people who have symptoms of AIDS is 365, so they don't overlap. So hopefully that's I don't know if that's gonna come up in a further question or if that's just interesting Let's keep going. A test sample of 500 millimeters of blood is taken from an HIV controller to determine the viral load. Tick one box that shows the number of virus particles that would be present in a test sample of blood taken from the median viral load. Okay. This is a unit conversion question. 500 millimeters cubed, okay? So look, 1 centimeter cubed is 10 millimeters by 10 millimeters by 10 millimeters. So there are 1,000 millimeters cubed equals 1 centimeter cubed. So 500 millimeters cubed is equal to 0.5 centimeters cubed. And this is given per centimeters cubed. So we need to do 212 divided by 2 because it's half a centimeter cubed. So the answer, 212 divided by 2, is 106. Okay? This was just checking that you can convert your units from one thing to the other. Let's keep going. All right, here we're going to use the data. Use the data in Table 3 and your knowledge of the immune response to suggest why HIV controllers do not develop symptoms of AIDS. Okay, so this is where we need to go back to this kind of table up here. So I said use the table and your knowledge. So we have to do both to get the 3 marks. First of all, let's start with the table. So I would start by saying that the viral load of HIV controllers is much lower, and in fact that even the highest viral load out of all the HIV controllers was still substantially lower than the lowest viral load of anyone with AIDS. Then I'd also talk about CD4 cells. The number of CD4 cells in the HIV controllers was much higher, and even the lowest was much higher than the highest CD4 count of people with AIDS. Now we need to use our knowledge of the immune system. So T helper cells are an important part of the immune system, and they help activate naive B lymphocytes, causing them to differentiate into plasma cells. They also help activate cytotoxic T cells, which help destroy cells that are infected with viruses. So they are a key part of the immune system. Let's look at the mark scheme. Okay. So they have just 1 mark for just saying they have more T helper cells and they have lower viral load. Okay, I'm not sure if I would have said that. Lower viral load to infect or destroy the T cells. This is what we're talking about. So I was just talking about activation of B cells or cytotoxic T cells or phagocytes, any of those 3. If you're talking about B cells, you could then go on to say production of plasma cells and antibodies. And if you're talking about T cells, the ability to kill virus-infected cells. So therefore, they're more able to destroy other microbes or even destroy mutated or cancer cells. Okay, on to question 6. So here we go. Scientists investigated the cell cycle in heart cells taken from mice 6 days before their birth and then at 4, 14, and 21 days after their birth. Day 0 is the day of birth. Here are the results shown in Table 4. So before we even get into the question, let's have a look at the results and kind of just try and figure out what's going on. So before they're born, there is a high percent of cells undergoing mitosis, and then that decreases quite substantially after they're born. It just gets less and less and less. What's going on over here? Percentage of heart cells undergoing DNA replication. Again, it's high and falls. That makes sense because in order for cells to go through mitosis, the DNA must first have been replicated. So we might be thinking about the cell cycle. I'll throw an image of that in here. So if a cell is going to proceed through mitosis, it has to go through that S phase of DNA replication, which is within interphase. That happens before the cell then goes through mitosis. So that's really what we need to explain. So describe and explain, 2 command words, describe and explain. It's only 2 marks, but I think we might want to say 4 things just to make sure. So we first of all describe the percentage of heart cells undergoing mitosis. It starts high at 13.9 and it falls dramatically over time, reaching 0.6. And then describe this as well. Starts at 8.5 and it falls dramatically, reaching 0 at day 21. I think we should probably reference that. It reaches 0. And then we would talk about this. This is because in order for cells to go through mitosis, DNA must first be replicated. And we might also say that the mice are growing rapidly before they're born. So if they're growing rapidly, then they're going to be doing a lot of mitosis. Let's check the mark scheme. Okay, the trend of slowing growth or trend of decreasing percentage. Yeah, okay, so that's the describe. You could have also talked about the DNA replication. It's the same mark point. And then the explain bit is this second mark point. The DNA replication has to happen before mitosis. Yeah. You could have talked about potency. The cells have lost the ability to divide. So heart stem cells becoming more differentiated. Could have talked about that as well. All right, let's move on. Next part of the question. The scientists determined the percentage of heart cells undergoing DNA replication by using a chemical called BRDU. Cells use BRDU instead of nucleotides containing thymine during DNA replication. Describe how BRDU would be incorporated into new DNA during semi-conservative replication. Right, I like this question. Now this question is one of those questions that might throw people, but is actually simpler than maybe you might initially think. So what might throw you is this BRDU. This is something that you've never heard about before. So that means that this question is an application question, but it's really just a content question in disguise. Because it says here, cells use BRDU instead of nucleotides containing thymine. So really you can just write about DNA replication, except when you would say thymine, just say BRDU instead, because BRDU is just taking the place of thymine. So I've dropped in a picture over here so we can quickly discuss. So what would we need to say? We're going to need to say all the normal things. We need to talk about, first of all, I would talk about helicase, breaking the hydrogen bonds between the two strands. Then we'd want to be talking about our nucleotides flying in. Okay, so our free nucleotides lining up. You actually don't need to know about the RNA primer for your spec, so we could kind of ignore that. But we talk about the free nucleotides flying in. Whoops, they're large there. And then, slight error there. The free nucleotides fly in and line up via complementary base pairing, but it is the DNA polymerase enzyme that joins the backbone. So what you should say here is that the BRDU nucleotides would line up and form a complementary base pair opposite an adenine nucleotide, because remember they take the place of thymine. T and A normally bind, but in this case it's going to be BRDU lining up opposite A, and then the DNA polymerase enzyme will join that to the backbone. Okay, so let's have a look at the mark scheme and see what they're kind of wanting from us. Are there any more marks? Just those ones there. Okay, so yeah, the helicase, breaking hydrogen bond, that's kind of exactly the same as normal DNA replication. This is the extra bit, the kind of application-y bit. The BRDU would be complementary to adenine, and you do specifically need to say adenine. You could also say that it forms hydrogen bonds. The DNA polymerase joins the nucleotides and incorporates BRDU into the new DNA strand and phosphodiester bonds forming. So really this is just a DNA replication question. It's just this bit is a little bit different. Oops, it's a marker. And this bit is a little bit different. Okay? Yeah, I just wanted to highlight this bit. This is a common misconception here. Okay, this is a common misconception. So hopefully you didn't say this. Reject if DNA polymerase catalyzes the complementary base pairing. Okay, so DNA polymerase does not bring the nucleotides in to pair up. They just float in on their own. DNA polymerase just joins the backbone. Okay, let's continue. Cells with BRDU in their DNA are detected using an anti-BRDU antibody with an enzyme attached. Okay, automatically, already I'm thinking antibody, enzyme, is this ELISA kind of stuff? Oh, And it is, because I didn't even read the next sentence, but there it is. Use your knowledge of the ELISA test to suggest and explain how the scientist identified the cells that have BRDU in their DNA. Oh my gosh. So this is a tricky one. So we don't know what type of ELISA test this is, or if it is even an ELISA test. And I'm wondering, so normally in an ELISA test you have some sort of fluid that's washed over like a stick, like a pregnancy test is a type of ELISA test, but I don't know if that would work here. What I'm thinking is they would probably just have the cells in a little dish, the cells which have got the BRDU in their DNA, and then we just apply the antibody, and then so we'd apply the antibody to the cells. Maybe we'd have a tissue culture of the cells, we'd apply the antibody, allow it to stick or not stick, wash it off, and then we'd add a substrate that produces a color change. So normally we have a colorless substrate that when the enzyme acts, it makes it blue. I don't know, it's quite often blue. I don't know why it's always blue, but like a colorless substrate that will then become blue, for example, enabling us to identify the cells with the BRDU. I think that would get the 3 marks. We'll look at the mark scheme in a second, but I'm wondering if there'd have to be something else, because I don't know if the antibody would actually be able to get into the cells to bind to the DNA. It would probably just be blocked by the cell membrane. Let's have a look at the mark scheme. Let's see if that would do it. So yeah, okay, it is. That is what we're talking about. Add the antibody to the cells, wash to remove excess antibody, add substrate to cause color change. Okay, yeah, so a tricky method to actually give detail for, but those are the basic points that we should say. Add the antibody, wash to remove the excess antibody. I don't know if I emphasized that enough when I was describing it. And then you add the substrate to cause the color change. So this wash to remove the excess antibody means that any antibody that's just sort of floating around and isn't attached to the cells will be washed away, and that's an important step. Okay, so that's ELISA. On to question 7. So Ulva lactica is an alga that lives on the rocks on the seashore, and it is regularly covered by seawater. Here's a diagram of it. We've got a kind of thallus, a bit like a leaf, and a holdfast that attaches the algae to the rock. So that might be important for kind of keeping it anchored in the face of waves and stuff like that. Maybe that's going to come up later on. Unlike plants, Ulva lactuca does not have xylem tissue. How is it able to survive? Well, this is a suggest question. Hopefully this appears to you quite quickly, but if you're struggling, always go back for a suggest and read the information at the top of the question. It says it right here. It is regularly covered by seawater. Because it's in the seawater, it doesn't need a water transport tissue. It just takes in enough water. I think we might need to say by osmosis from the sea, possibly. Let's quickly check the mark scheme. Let's see if that's what they want you to say. No, that's not it. You're not going to get that. So short diffusion pathway. I guess, yeah. I mean, it is flat. It has a surface permeable to water or ions. Yeah, I would not have got that. So, I mean, surface is permeable. I wouldn't have said it this way. Short diffusion pathway. And because it is surrounded by water, I almost feel like they should say short osmosis pathway, because it is osmosis, not diffusion. But yeah, so you also need to recognize in the picture the fact that it's got a very large surface area, it's flattened. Okay, interesting one. Moving on, Ulva lactica has a haploid and a diploid form. Figure 7 shows the life cycle of Ulva lactica. Okay. On Figure 7, complete each box with an appropriate letter to show the type of cell division. T to represent mitosis and E to represent meiosis. Okay, so we just basically need to look at the diploid and haploid bits. Diploid to diploid, that is a mitosis. Diploid to haploid, this has got to be meiosis, so that's it. We'll put it as an E. haploid to haploid, that also has to be mitosis, okay? Because we're not changing the number of chromosomes. It's an identical thing, haploid to haploid, so that's also mitosis. Haploid to haploid, that's also mitosis. And then when these things, this is fertilization, is happening here, and then we get the zygote. Interesting. Okay, 7.3. Oviprolifera also produces haploid mobile single cells that can fuse to form a zygote. Suggest and explain why successful reproduction between oviprolifera and ovilactula does not happen. Okay, so I mean, I got ideas for why this might not happen. It might be something to do with differing number of chromosomes, so that when fertilization happens, we can't get the formation of homologous pairs. But I think we also need to say something which you might overlook, an obvious thing, which is if you look at these two names, these are different species. I think this is testing your knowledge of binomial names. Sometimes you have to think, what is the question trying to test me on, to think about what's in the mark scheme. So Oviprolifera and Ovilactuca are different species, and therefore fertilization between the gametes would not happen. And then potentially because they have different number of chromosomes, so we couldn't form homologous pairs, is what I would possibly go for. Let's see if that's what they want. Okay, so they are different species, is worth a mark. So if fused together, if fused together, they would not produce fertile offspring. We've got to also remember the definition of a species. A species is something that can form fertile offspring. You can kind of explain that in a few different ways. Here's just accepting these things. If fused together, meiosis could not occur. If they fused together, chromosomes could not form homologous pairs. So that would have just about been allowed. But this is the definition for a species, which is kind of what the question was testing. Okay, 8. The water potential of leaf cells is affected by the water content of the soil. Scientists grew sunflower plants. They supplied different plants with different volumes of water. After 2 days, they determined the water potential in the leaf cells by using an instrument that gave a voltage reading. Okay. The scientists they've got a calibration curve, so we can convert a voltage reading into a water potential. Never seen this before. This is interesting. Okay. The scientists needed solutions of known water potential to generate their calibration curve. Table 5 shows how to make a sodium chloride solution with a water potential of minus 1.95 megapascals. Complete Table 5 by giving all headings, units, and volumes required to make 20 centimeters cubed of this sodium chloride solution. Okay, so we need to so we kind of ignore this. We just need to focus on this, to be honest. Okay, I was a bit confused there for a minute. The concentration of sodium chloride solution that we need is this, 0.04. So really the question is asking us, how can we make, given 1 mole per dm cubed, sodium chloride solution, and if we want to make up 20 cm cubed, then how do we do that? So this is 4% of this solution, and we want to make up 20 cm cubed. Okay, so over here we're going to have the sodium chloride solution. We're also going to have volume, and it will make you write it out in full, otherwise you won't get the marks. I know it's a bit annoying, but volume of distilled water. We need the units here, and this is cm cubed. This is cm cubed. So we would need, I'm going to get my calculator out, we need 4% of 20 cm cubed. So we need 20 times 0.04. 20 times 0.04 equals 0.8 centimeters cubed of this. And then the rest of it, to make up the 20, has got to come from the distilled water. So that's going to be 19.2 cm cubed. Okay? All right. Double check. Yeah. Yeah. Good. Fine. Okay, let's keep going to the next question. Table 6 shows some of the concentrations of sodium chloride solution scientists used and the water potential of each solution. There is a linear relationship between the water potential and the concentration of sodium chloride solution. Use the data in Table 6 to calculate the concentration. Okay, it's a linear. Oh my god. Use data tables to calculate the concentration of sodium chloride solution with a water potential of minus 3.41. Oh, Christ. Sorry. Okay, so this goes up by 0.6. This goes up by 0.06. Sorry, this goes up by 0.2. Okay, so this is like a y equals mx plus c, I think. Kind of maths question, which is super annoying. So the water potential, so we convert. Oh my god, I love the maths. Minus 1.95. Okay, that's this value. Y equals m times x, which is this value, 0.04. plus C, and then we've got another set of data. So okay, we need to find out the m, okay, the gradient, the slope. So to do that, we basically look at the change here versus the change here. So the change here is 0.06. Yeah, that's the change. And what's the change in water potential? We do 4.87. 4.87. It's needed minus. Minus 1.95. Minus 1.95. So the change. Oh, hang on a minute. I went wrong. 4.87 minus 1.95 equals. Okay, so that's 2.92. So the drop here is minus 2.92. So then m is equal to minus 2.92 divided by 0.06, which is equal to 48.67. Yeah, okay, so now we need to solve the equation. What's C, though? So now we need to put those numbers in. So minus 1.95 equals 48.67 times 0.04 plus C. Therefore, let's do that. So times 0.04 equals minus 1.95 equals 1.95 plus C. Okay, so C is 0. Amazing. Basically, C is pretty much close to 0. So therefore, we can just, now we're trying to work out minus 3.41. So we just do minus 3.41 equals, this was M, 48.67 times X. So then we just divide both sides by 48.67. That should get us on the answer. 3.41 divided by 48.67 equals 0.07. That took a lot of work for 2 marks, I think, but I hope that's the right answer. Maybe you found a better way of doing it, but the clue for me in the question was this linear relationship thing. So therefore, you do need to be able to do this. Okay, this is GCSE maths that you need to be able to do. I haven't done GCSE maths for quite a while, so that took me a while. Let's double check it's the right answer. Hopefully it is. Yeah, correct answer of 0.07. And then any evidence of this gets you a mark. So that's that M bit, like y equals mx plus c. So working out what m is gets you the mark. Okay. Tricky one. Let's keep going. In addition to determining the water potential in the leaf cells, the scientists measured the growth of the leaves. They recorded leaf growth as a percentage increase of the original leaf area. Okay, here are the results. So looking at the water potential and how the leaves grew. Okay, one leaf with an original area of 60 centimeters squared gave a voltage reading of -7 microvolts. How much did this increase in area in centimeters squared? Oh, they love maths in this paper, don't they? There's so much maths. Okay, so this is the figure that we've got to get first, -7 microvolts. We want to turn that into a water potential, so we go up to figure 8. -7 on figure 8. Here we are. Right, zoom in. Make sure that this is accurate. -7. This is -5, this is -6, this is -7 right there. We follow that all the way along to this point here, and then we're going to follow it all the way down to that point there. So that is -1.9. -1.9, I believe. So -1.9 is the water potential in megapascals that we want to use down here. So now we're looking at -1.9 megapascals, and that is actually bang on there, isn't it? Because this is -1.2, 4, 6, 8, 9 is exactly that figure there. So that's a 15% increase. Okay, so now we have a 15% increase. And we now want to know what that is in actual centimeters squared. So it says here that the original leaf area was 60 centimeters squared. So we want to do what is the increase of that. So we do 0.15 times 60 to get the actual increase in centimeters squared. 0.15 times 60 is 9 centimeters squared. Okay, so that question was tricky because you had to look at multiple graphs and kind of just keep an eye on what you were actually doing. Let's check the mark scheme, check I got it right. I hope I did. 9, yeah. But you would get some marks if you didn't get all the way for evidence of calculating a water potential, so that's using the top graph, or somehow getting evidence of knowing the growth is 15%, or evidence of having calculated that it would go from 60 to 69, but then if maybe you forgot to, like, because it's actually asking for the difference, not the new figure. Okay. On we go. 8.4. Still on question 8. Sunflowers are not xerophytic plants. Xerophytic plant, this is something like a cactus, a plant that is adapted for dry environments or habitats. The scientists repeated the experiment with xerophytic plants. Okay, so the first bit of this experiment I'd actually forgotten was all about sunflowers, was it? I'm going to go back up to the top. Okay, so the first bit about this, the whole experiment up till now has been about sunflower plants, not xerophytes. But now we're going to talk about xerophytes. Suggest and explain one way the leaf growth of xerophytic plants would be different from the leaf growth of sunflowers. In figure 9. So we need to talk about figure 9. Okay, I was slightly wondering what they were wanting me to talk about, but in figure 9, so this is figure 9. This one here. So we're kind of just saying, how would this be different for a cactus, for a xerophytic plant? So we might say that a xerophytic plant, there's less water around, so maybe the water potential of the cells is more negative because the solution inside the cells is more concentrated. They're just less, it's less pure water, it's more concentrated solution. So I would imagine that this line, instead of going like this, maybe it would extend further this way, like that. So just like it would have a sort of similar shape, but the plants could just tolerate a lower water potential and still grow reasonably well. So that's how would I describe that in words? It says suggest and explain. So suggest, I would expect that the plants could grow at a rate of 15% up into a greater water potential. I might say the graph might look similar, but reach 0% increase in original area at 5 megapascals instead of 4 megapascals. Explanation, because xerophytic plants are adapted to environments where less water is available. I would have got 1 mark for that answer because I didn't explain it enough. Okay, so I would have got this mark. Growth may continue at lower water potentials. Speculating as to why. How would you kind of speculate as to why? I said adaptations, but I think you would have to be specific. So adaptation in enzymes, or adaptations in photosynthesis, or adaptations in metabolic reactions. Probably enzymes would be the easiest thing to kind of remember. So if you are talking about general adaptations and you're trying to make it more specific, say adaptation in enzymes. That could apply to lots of different adaptations, I guess. You could have also approached this question from a totally different angle and said that xerophytes would have had just slower growth, so not up to 15% in change in leaf area, because there's a smaller number of stomata for gas exchange. And if you did talk about that, you had to talk about stomata. Okay. 8.5. Use your knowledge of gas exchange in leaves to explain why plants grown in soil with very little water grow only very slowly. So you need to remember that xerophytic plants have much fewer stomata than non-xerophytic plants, which means that they have less ability to take in carbon dioxide and therefore less ability to produce their own glucose and carbohydrates. So they're going to grow less quickly because they have a lower rate of photosynthesis. Let's do that again. Okay, so you need to remember that xerophytic plants have far fewer stomata, or they might even have sunken stomata, compared to non-xerophytic plants. With fewer stomata, or stomata that are closed more often, we're going to have a reduced rate of diffusion of carbon dioxide into the leaf, so therefore a lower rate of carbon fixation and photosynthesis, a lower rate of glucose synthesis, and a lower rate of growth. Let's check the mark scheme. Less carbon dioxide for photosynthesis. Stomata close. So this I feel like is a bit harsh, stomata close, because it might just be they have fewer stomata, not that they're closed. But you did have to say the stomata close. Why is that exactly? Let me just look back at the question. Oh, okay, because it's talking about plants. It's not talking about only xerophytic plants. It's talking about general plants. So actually, it wasn't really talking about xerophytes. It was talking about generally. And yeah, generally, if a normal plant has less water, then it's going to close its stomata in response to that water stress, and that's going to slow its rate of growth. Okay, question 9. Scientists investigated the affinity for oxygen of horse hemoglobin and mouse hemoglobin. Some of the results are shown in table 7. Horse and mouse here, partial pressure of oxygen at which hemoglobin is 50% saturated, partial pressure at which it's 25% saturated, and then the body mass of an animal. Okay, plot the data and use these points to sketch the full oxyhemoglobin dissociation curves for a horse and a mouse. Okay. So first of all, we need to find out what is the biggest partial pressure we ever need to plot, and that's 6.5. That's at 50% saturated, so I think we're going to need to go. Normally the shape of a curve is a bit like this, right? So if it's at 50% saturated, I probably want to double that to get to the top. So I think I probably want to go up to 12. So that would make my axis down here something like 2, 4, 6, 8, 10, 12. Yeah. 2, 4, 6, 8, 10, 12. All right. Percentage saturation of oxyhemoglobin. This is more simple. This is just going to go to 100%. So that's probably going to go 20, 40, 60, 80, 100. Notice that when I'm doing this, I kind of trial it out before I write anything. I'm going like 20, 40. I'm just checking in my head that it's all going to fit. You should always do that. Don't just start writing. Kind of just check with your pen or pencil before you start writing it down. So that's the kind of that's the scale done. All right, next thing. Let's plot the horse. So 3.2 for 50 for the horse. So 50 is here, and 3.2. So that's going to be 3, and then 0.2 is there for the horse. And we also want to do the 25%. So that's 1.9. So 1.9 is 1.9, 25% is going to be right in there, and 1.9 is going to be, that's 1.8, so 1.9 is going to be there. Okay, and now we want to try and sketch that graph for the horse. It's going to be sort of extremely, it's going to be 0, 0. It's going to be something like, and then I'm going to want to keep it going, and it's going to tail off to 100. Oops, that was not a very good curve. Do you want? If I actually was doing this, I don't think I can't do a ruler on my screen here, but it's not quite a straight line. It is. You do want to have it sigmoidal slightly, but then it's going to curve to 100. Let's just say 100. All right, let's do the. What's the other one? The mouse. Okay, so we want 6.5 and 3.3. So 6.5 for 50. So 50 is going to be at 6.5. That's going to be right there. And then 25 is going to be 3.3. So that's 3.3 is going to be there. So we're going to do kind of sigmoidal curve slightly, and it's going to taper off like that, I guess. Okay, hopefully that will give me the 3 marks. Let's see what the mark scheme looks like. I wonder if they're going to give us an example graph. Okay, so we need the x-axis with the y-axis 0 to 100, and the x-axis minimum 1 to 8, but could be bigger, and use at least half the size of the grid. Correct plots, both curves leveling off at around 100%. Okay, cool. So that works. What I drew meets that criteria. All right, moving on. 9.2. Wow, more maths in this paper. The following equation can be used to estimate the metabolic rate of an animal. 63 times the body mass in grams to the minus 0.27 power. Okay, so we need to do this for mouse and a horse. So the mouse has a mass of 23 grams, and the horse has a mass of 550,000 grams. Okay, so we need to basically just use this equation twice. All right, so MR, I'm going to put MR mouse equals 63 times 23 to the minus 0.27, which equals 63 times 0.428, which is equal to like 27.02 for my mouse, and then I'm going to work it out for the horse. Let's say horse metabolic rate, right, this way around, metabolic rate horse, that's going to be 63 times body mass, which is a massive number, 550,000 to the minus 0.27. So 63 times 0.028 times 68 equals 1.7. I'm going to do it to 2 decimal places, 1.78. Okay, and then it says, how many times faster is the metabolic rate of the mouse than the horse? This is the mouse than the horse, so I want to do 27.02 divided by 1.78. So this big number divided by this smaller number. So that is going to be 27.02 divided by 1.78. I've got 15.2. I'll probably give it to 1 decimal place. I've got 15.17. 15.2 times faster. Right, let's see if that's correct. Okay, yeah, so somewhere between 15 to 15.4. All right, let's keep going. 9.3. The data in Table 7 show differences between the oxyhemoglobin dissociation curve for a mouse and oxyhemoglobin dissociation curve for a horse. Suggest how these differences allow the mouse to have a higher metabolic rate than the horse. So we need to look at our graphs again to kind of understand what this is going on about. Well, okay, so the main difference is that there's a steeper gradient for the horse compared to the mouse. So that means the horse is more fully loaded at lower partial pressures of oxygen, whereas the mouse has kind of a more is not fully 100% saturated with oxygen at sort of intermediate partial pressures. So I think this means that maybe the mouse can unload oxygen more readily, I think is the phrase we'd have to use. So that we'd say that the mouse hemoglobin has a lower affinity for oxygen and can unload its oxygen at respiring tissues more readily, thus allowing a higher metabolic rate. 2 marks, that should do it. Let's check. Oops, that's the wrong paper. That's the one. Yeah, okay, good. So, mouse hemoglobin has a lower affinity, and therefore more oxygen can be unloaded for respiration in respiring tissues. Okay. Pause there if you want to have a look at anything else you could have said, and then we'll continue. Okay, mammals such as a mouse and a horse are able to maintain a constant body temperature. Use your knowledge of surface area to volume ratio to explain the higher metabolic rate of a mouse compared to a horse. Okay, so we're definitely going to need to expand on this idea of surface area to volume ratio. So mouse, small size, high surface area to volume ratio, and then a horse is going to be large size, low surface area to volume ratio. Hopefully that will get us the first mark. And then we need to talk about, with higher surface area to volume ratio, we're going to lose heat energy rapidly. And with a lower surface area to volume ratio, we're going to retain more or lose less heat energy compared to the horse per gram of body mass. So the higher metabolic rate of the mouse is because metabolic reactions need to run very quickly to maintain body temperature. Okay, let's check to see if that is, or what language we need to use. So for the mouse, larger surface area to volume ratio, faster heat loss, and then a greater rate of respiration or metabolism to release heat or produce or generate heat. There we go. Pause if you need to, and then we're going to move on. Okay, question 10. Finally, we've made it to the last question of paper 1. Remember, these questions on question 10 are normally like fairly straightforward content questions, AO1. So properties of water. I actually think this is one of my big predictions for what might come up in paper 3 this year. I feel like this could be expanded into an essay. So if you're one of my students, you should know this pretty well. Here are all the properties. I'm just going to quickly talk through them. So first of all, it's a metabolite in condensation hydrolysis reactions. For example, forming glycosidic bonds between 2 glucose molecules is a condensation reaction, or it can be used in hydrolysis to split, let's say, maltose into 2 glucose molecules. It's a solvent in which metabolic reactions can occur, or a solvent to transport substances around the body. So blood is mainly water, and so is the xylem and the phloem sap. It has a high specific heat capacity, so it resists or buffers changes in temperature. By the way, the importance of that is that it maintains relatively constant thermostable internal environments inside the body. It also has a large latent heat of vaporization, and this is very important in cooling. So when we sweat the water on the surface of our skin, as it turns into water vapor, it removes heat energy from the skin, cooling us down if we've overheated through exercise or whatever. There is also cohesion between water molecules. That's the kind of stickiness of the water molecules, and this supports the column of water. What that means is that when water evaporates out the top of the leaves, it kind of pulls up on the water molecules below, so they stick together. So when you remove water molecules from the top of the column, ones below are pulled up through the hydrogen bonding network that is cohesion. This phrasing here, let me just highlight here. If you ever get a question on xylem and transpiration, you've got to say this, the cohesion tension theory. This is like really key language that they want you to use normally. Also, you could say that cohesion is useful for producing surface tension to support small organisms, like a pond skater can sit on the surface tension of the water. Extra points, AVP, down here, ice floating to maintain an aquatic habitat beneath, also water transparent to allow light to penetrate down to the bottom of a pond, a shallow pond, to allow photosynthesis in plants growing in the pond. Okay, pause there if you need to. Oops. Pause there if you need to, and let's move on. Okay, describe the biochemical test you would use to confirm the presence of a lipid, non-reducing sugar, and amylase in a sample. Okay, so lipid is fairly straightforward, the lipid test. Non-reducing sugar is a little bit more convoluted because it's a Benedict's test, but you have to first do a hydrolysis step. And amylase, you might think, this is an easy one to throw you off course, because you might read it as amylose starch, but it's not that. Amylase is an enzyme, so this is a burette test for a protein. Okay, let's just look at the mark scheme, and I'll kind of illustrate with some pictures as we go. Okay, so here we have the biochemical tests. First of all, the lipid test. So first you add ethanol to the food sample, that's shown here, and then you shake it up and you mix it really well. That actually dissolves some of the lipids in the ethanol, and then you add water. When you add water, that kind of dilutes the ethanol, and it means that some of the lipid will come out of solution as a milky white emulsion, which generally, if you leave it, will rise to the top, or it just might make the solution cloudy. So that's the lipid test there. Remember those steps. Right, the next test is a bit more complex. So remember, this is the test for a non-reducing sugar. So actually, the only way that you can test for a non-reducing sugar, or the presence of a non-reducing sugar, is to first do the Benedict's test to see if there is a reducing sugar. And we kind of have to just rule it out. So we just make sure that there isn't any reducing sugar there, first of all. Then we go back and we do this acid hydrolysis step. So this is the key acid hydrolysis step. So we boil it with an acid and then we neutralize it with an alkali. You can give an example of an alkali, I think down here, sodium carbonate or sodium citrate would be alkalis that you could use, or sodium hydrogen carbonate. Yeah, those are named alkalis that you could use, even though they're also in here. And then you have to heat it with Benedict's again, and if it's positive then we know that there is a non-reducing sugar. So these marks here were quite tricky to get. You do also have to give the red-orange precipitate the result of the test. Finally, amylase. You needed to remember that amylase is a protein. We're not looking at amylose starch, it's amylase. So this is burette reagent and it becomes purple. Or, look, there's another option here actually, because it is an enzyme, we could actually test, that's an interesting idea, we could test by adding starch and then seeing if it gets digested. So add starch, test for the starch, and then leave it for a while and then test for the starch again. That's an interesting idea. Okay, let's move on to the final question. All right, describe the chemical reaction involved in the conversion of polymers to monomers and monomers to polymers. Give two named examples of polymers and their associated monomers to illustrate your example. Okay, this is brilliant. Loads of marks available here. I'm just going to drop in a table. Okay, so first of all, before we get into specifics, I would talk about general condensation reactions and hydrolysis reaction. So I'd say reactions that join monomers together are condensation reactions, and they produce a molecule of water when a new covalent bond is formed. And I would say reactions that split a polymer up into its monomers are hydrolysis reactions. They require a molecule of water to go in and break the covalent bond to separate the polymer into its monomers. Then we could talk about specifics, named examples. So maybe glucose molecules into a carbohydrate such as starch, condensation reaction forming glycosidic bonds. Or we could talk about starch being broken down by the enzyme amylase, which breaks the glycosidic bond and produces, well, it actually produces maltose disaccharides. Lipids and fatty acids, we're talking about glycerol and fatty acids joining together in condensation reactions to form lipids, and it's the formation of an ester bond here. Nucleotides are the monomers that can build up to form a nucleic acid, for example DNA, and we're talking about phosphodiester bonds forming in a condensation reaction. We don't have many of these. We don't really talk about this on spec, but there is something called a nuclease enzyme that takes big nucleic acids and breaks it down into the monomers. Amino acids, the proteins, we're forming peptide bonds as a condensation reaction, or we can hydrolyze them, protease enzymes. So that should be easy marks, really, I think. So let's just look. So in terms of the overall kind of explanation, we need this. A condensation reaction joins monomers together and forms a chemical bond. You could have said covalent bond, I think is fine, and releases water. Okay, all of that is needed for that general point. You've got to say those three things, and that is something worth remembering. I've seen that come up in quite a few times. Condensation, chemical bond, and releases water. Similarly, this kind of other general point, hydrolysis reaction breaks a chemical bond between monomers and uses water. It's kind of the opposite process. And then we have example and reference to the correct bond. All right, I hope you found that useful. I certainly found that quite a challenging paper, especially the mathematical content and some of the tricky language that AQA requires you to use. I'm still getting used to the AQA spec. I've been teaching it for about 18 months. and I've got about 15, 16 years experience of OCR, so I'm still learning the kind of detail that they want you to include. And you should be doing the same too as you gear up for your exams. Absolutely important thing that you do is look at how many marks you got per question. And if you got a low number of marks in a specific question, you need to go away and revise that specific topic or skill. Was it content knowledge that let you down, or was it a mathematical skill that you can practice by finding some questions with that skill repeated many, many times? All the best in your revision, and good luck with your exams.